the characterization of a particular biological process. In our case, we are a lab dedicated to the study of the function of the ubiquitin system in plants, using mainly Arabidopsis, although we are extending those studies to other systems. Actually, we want to move also to Mercantia to start characterizing the function of that ubiquitin proteasome system in the early land plants and how this system has evolved. So I will show that, I mean, some part of that, the research that we carry out in the lab in the second part as a case of a study in which we apply different proteomics or protein-protein interaction techniques in our daily research. And so, as I said, so maybe this first part will be too basic for many of you. And well, but I think it's a good idea to start refreshing those concepts and those terms. And in the end of the talk, I will move to actually the proteomics tools or techniques that we use to identify protein complexes, particularly from Arabidopsis in our lab. But also, as I said, we are moving also to other systems. Okay, so and just also to introduce my colleague Monica. So after lunch, she will talk about, she will show you how we handle the MS data we obtain from our proteomics facility, and to filter and to discriminate or select those proteins that are really significant, and how to categorize them, organize them, and identify those that are really interesting for our research or that can open new avenues of research in our lab. Okay, can you hear me so correctly? So first, so is everything fine?
Yes, everything is fine.
Because the connection today in our institute was kind of trembling, and so if you stop hearing me, so please let me know, because I can keep going on and maybe for 5 minutes I am alone. Okay.
Okay.
Okay. So see how to. Okay. So yes, as I said, some terms about proteomics. This is general, not just for plants. The proteome is a complement of a genome. And this is a very simple definition. Actually, what we want to do with proteomics is to study in a high throughput manner the expression and function of all proteins in a genome, no? In a particular organisms. One thing I will also, so this presentation will be available. So just you, I mean, can relax and just go through it easily. So right. So and then about why we study the proteome. So well, because the DNA sequence does not reveal all the biological function by itself, because one particular gene can produce many different products at the protein level, because RNA splicing, I mean, alternative RNA splicing can occur. So different isoforms of the same protein can be produced. Also because even the genome is, same genome is present in all cells from an organism. So the proteins that are produced there can change a lot depending on the tissue, the cell type, the developmental stage, or in response to different environmental cues, okay, or stress responses. And so the slides are moving fine. So is it? So can you see me changing slides? No? Okay.
Yes.
Okay. So another issue when we study the transcriptome versus the proteome is that there is a poor correlation between the levels for both biochemical entities. No, what we have observed is that the protein level or the abundance for a particular protein could, I mean, be completely unrelated to the levels of its own transcript, and that's because there are many post-transcriptional mechanisms that control the stability of or the processing of a specific transcript, and also post-translational levels that will change the abundance of that the particular product of that transcript. No, and also another consideration is that a transcript can produce many different isoforms for a protein depending on the post-translational modification occurring on that protein. And so this is also very, very basic and generic. So you know that there are many different post-translational modifications happening in different proteins, and that is going to change their function, their stability, or their ability to interact with other proteins or to localize in different subcellular compartments. No? And these are some examples, not just for plants, but you can see here many different post-translational modifications that have been reported. As I said, there are about 200 different ones. And what we want to do with proteomics is to characterize different issues on this protein. So we can make a classification of proteomics depending on what we want to study the function, the expression, the abundance of those proteins, and also the structure. So today we will focus in interactomics, which is related to functional proteomics. But as I said, all these three different types of proteomics complement genomics and transcriptomics to study in a general omic view of the functions of an organism, or even at the cell specific level. So to carry out this proteomic approach, we need high throughput tools that rely on different levels, molecular biology tools to extract or to collect all proteins from an organism's tissue, cell type, different tools to separate those proteins and display them, then the identification tools, for example, mass spectrometry, and also, although I will not talk about this, but proteomics can be used for structural, from a structural point of view, no? to characterize the structure of a particular group of proteins. As I said, there is a classification of different types of proteomics, expression proteomics, where we will study quantitative changes in the abundance of the whole proteome of an organism's tissue, cell type. Differential post-translational modifications can be also analyzed in a high throughput manner. And we can even characterize the proteome of a specific proteome of a subcellular compartment, the nuclear, membranes, organelles. So for functional proteomics, which is actually the focus of this talk, we are aiming to characterize the function of a specific protein, the activity, and also the ability to interact with other proteins or to become part of a protein complex. And finally, as I said, there is also structural proteomics. And for interactomics or functional proteomics, protein-protein interactions in the protein level, so we want to unravel the biological roles of proteins in clusters or functional pathways involving particular cellular mechanisms. So these are all definitions so that I will keep in the presentation. And a few considerations when we work with plants. So one of them is that the genome sequence that we actually require to identify, afterwards you will see all proteins analyzed or proteins identified using the different techniques I will show afterwards. So the genome sequence is not available for all plant species that we work usually. So although the number of genomes that are available is increasing, at a very fast pace, which is really cool. But indeed, whole genome sequences are required to carry out an accurate proteomic study. Then plants are always challenging when we want to extract proteins from them. And also it changes or it varies from plant species to plant species. So, I mean, many protocols have been set up for Arabidopsis and rice and maize, but it doesn't mean that it can be easily transferred to other plant model. And because I mean the particular chemical or physical properties of those I mean the cell compartments or cell walls in those plant systems. So it means that when we start working with a different model, we need to set up again many steps in the purification protocols or processing protocols. And as I said, so this is connected to the next one. So the methodology needs to be updated depending on the plant species. So, but well, fortunately, many protocols are already available for different plant species that we can apply, particularly for Arabidopsis. For functional proteomics, there are different separation and display tools that we can use to screen for interactions with our protein of interest. No, they can be classified into different types. Those that allow detection of binary interactions, mainly, so for example, the yeast to hybrid screens, or protein arrays, protein chips. That in specific cases can be combined with, I mean, can involve additional proteins. So I mean, we know, of course, yeast-3 hybrid systems that allow detection of interaction of trimeric complexes. But yeah, these two techniques have this significant limitation that can be overcome by other techniques like biochemical purification or affinity purification of protein complexes that allow this detection of multi-subunit protein complexes. So these are the ones that are more interesting, I guess, for the people, that the participants, and I will pay more attention to this, particularly the affinity purification systems. So just to fast overview of the other ones, because I could see that the participants, I mean, the audience kind of heterogeneous, so in their background, but I will go quickly through all these techniques because I believe that they are all pretty familiar with them. So yeast to hybrid analysis is based, I mean, it's performing a heterologous system. So in yeast, in which we will express two different fusions, no? Our protein interest fused to the activation domain of the usually GAL4 transcription factor, and the proteins to be tested will be fused to the binding DNA binding domain of the GAL4, and vice versa. So we can switch, no, these proteins, so to test in the two directions, no. And when these two proteins interact, where we get this induction of the expression of a reported gene. So usually it's the beta-galactosidase enzyme, or enzymes that allow synthesis of specific amino acids for which the yeast strain, which we are working, showing exotrophy. So this expression of this reported will allow the yeast cell to survive in a specific media lacking some specific amino acids. No? And that's why, I mean, how we can select specific yeast clones that will contain proteins that interact with our protein of interest. No? This is an example of a yeast to hybrid screen performed in our lab, actually by Monica, that we'll talk later, in which she screened yeast to hybrid Arabidopsis DNA library against her protein of interest, and she used two different reporter systems to identify proteins that interacted with her bait. No? And you can see here that there are yeast clones surviving, no, growing in the media with those exotrophies lacking these two amino acids, tryptophan and leucine, but also we can combine it with alanine and histidine for more astringent selection, and also with the 3AT compound that also restricts more the interaction. So allowing detection of very strong interactions, together with XGAL, a substrate for the beta-galactosidase. So the the stronger the interaction is, the more color you get. No? And here you see in the other part of the panel, so she afterwards organized all those clones and confirmed those interactions, no? Those by repeating the is to hybrid screen with a organized library. And upon selection of those clones, DNA extraction, transformation of E. coli, and sequencing of the plasmids that contain the proteins of interest. So it's possible to categorize or classify using gene ontology tools. So the different, the potential functions of the proteins that interact with your bait. Actually, well, she found several proteins that are related to transcriptional regulation, others that are related to nuclear metabolism of different functions that you can find more or less interesting. So particularly in our case, those related to transcription regulator were of special interest. So this is a blind screen. So you are just screening a library and see what interacts with your protein. You can do it all the way around. So just checking a collection of proteins that are expected to interact or that are part of a specific biological process. So this was a screen that I carried out while I was a postdoc using a specific protein. I will talk a little about it later, in which, we had a collection of cDNA clones in different yeast strains, and what we did is just to combine them, the clone, and see the potential interaction between them. So, and in this way, I identify how this protein was interacting with different components of this specific biochemical complex. And you can quantify this using the beta-galactosidase activity, particularly adding this substrate, the OMPG. And just by measuring absorbance, you can measure the strength of that interaction. Particularly this protein of mine was interacting strongly with DDB1, which is part of the same complex, and also with other proteins around there, so including some members of the COP19 allosome. I will talk about this in the second part of the talk. So these are examples of how to use these two hybrid. Yes, to screen, to carry out a search of potential interactors, or to look for interactions with a specific collection. So there are available collections of transcription factors that can be tested against your protein of interest. And yes, the other, as I said, system to look for binary interactions is to use protein arrays. This is maybe not so common now, not so fancy. It depends. Here, what we will try to do is to immobilize a collection of proteins into a platform. Here you have different systems and the advantages and disadvantages of them. So having the proteins in diffusion, like in a multi-well plate, or having them absorbed, or binding them using the covalent link to the surface. And this one supposedly is the very best one, to have the system that allows you to have the proper orientation of the proteins to be tested, so that they can interact with a library of proteins, and for to search for interactions, in the most appropriate way. And these chips or these arrays, protein arrays, can be tested not just to check for protein-protein interactions, to screen for protein-protein interactions, but also at the functional level, they can allow for detection of antibodies that interact with our particular protein, and also to characterize enzymatic activities. We can provide to those protein arrays different substrates, labeled with a specific probe, and detect which proteins in that array are catalyzing or metabolizing that specific substrate. Okay. So, so there are some of these chips that allow for quantification of the abundance of a specific protein. So we call expression array. and others that allow for protein-protein interaction screens in a high throughput manner. And finally, as I said, functional arrays that allow also to analyze enzymatic activities, no? And what you detect is the reaction products. So you can combine all these three, and you can get an overview of the function of many proteins in your system. This system has a limitation, is that you need to produce all those proteins. So, I mean, it has several limitations and advantages, but one of the most important limitations is that you need to clone the open reading frame for all those the proteins that you want to test, and this is really a challenge. It's time consuming, and well, actually, it also has other limitations. Is that you will produce just one open reading frame in a heterologous system from which you will purify your recombinant protein, and you are limiting the production of specific protein to usually one isoform. No? And this is, as I said, it's a limitation because you are missing a lot of information that could be identified by using different isoforms for the same protein. No? So in this case, what you would do is to express that particular protein, well, the collection of proteins, all fused to an affinity tag. In this case, is GSG, and another secondary one. So this is 6 repeats of histidine. That in this case, in this example, will allow immobilization of the resulting proteins to this nickel-coated platform. So what you get is like this collection of proteins, all organized in that platform. And for this, well, there are available robotic systems that allow, I mean, that facilitate all this arraying, in which actually what you do is to express all those proteins, usually from bacteria, in the multi-well plates, and purify and extract proteins right away in those plates. And after purification, they can be arrayed into those platforms that can be used for the specific screen that we want to carry out. Okay. One of the important things on this system is that usually the efficiency of purification, of expression and purification of a particular protein can change a lot from protein to protein. So then you need to measure the abundance of each spot in the array for a specific protein. Well, that can be accomplished using an anti-GST antibody, so labeled, and what you get would be this kind of image. So this is using the scanning of the scanner to visualize the abundance of each spot that has been labeled with the anti-GST probe. And you can see that there are changes from spot to spot. That well, actually, by measuring this fluorescence, so we can do relative quantifications once that we use the actual probe that we want to be tested, no, in the array. And so this is what we will do with our specific probe that can be our protein of interest labeled with a fluorescent marker, or as I said, these arrays can be used at the functional level to characterize enzymatic activity. So it can be a protein, the one that is labeled with the fluorescent probe, and will interact with specific proteins in the array, or what we can do is to add a substrate or a specific substrate that we want to test against a specific collection of proteins. No? For example, a collection of kinases or other enzymatic activities can be arrayed or organized in these chips, and we can test different substrates labeled with a fluorescent marker, and to see which ones are actually processing those substrates. No, that is what you can visualize here in this scanner. Okay, so those two techniques mainly allow for binary, protein-protein binary interactions, and have certainly certain limitations, no? Because we need to have a specific library of cDNAs. We need, maybe in the case of protein chips, to clone them, to express them, to purify them. So it's actually a challenge. So different systems have been developed to overcome these limitations and to purify proteins right away from cell extracts from plants. So the classical one was the biochemical purification of proteins or protein complexes using chromatography techniques. So these are based in different properties of proteins, so biophysical properties of proteins, and that allow a certain level of fractionation of those proteins from cell extracts. That fractionation will occur depending on their size, or their hydrophobicity, or their charge, or their affinity to the certain matrices. So this is the canonical classical way to purify protein complexes from different organisms. And this is an example that I could witness while I was a postdoc. This purification of a specific protein is the COP10 complex, purified by Yuki Yanagawa, so a number of years ago. And so in that case, so she, sorry, she purified this complex from cauliflower because it's a relative of Arabidopsis. So she was characterizing this specific protein COP10 from Arabidopsis. So, but as she needed a large amount of material, so she decided to work with cauliflower. And by different, steps of fractionation, of purification, different columns, so she ended up with a semi-purified sample that she applied to a gel filtration column to fractionate the protein depending on their size. And she collected fractions that contain her protein of interest, COP10, because actually she had an antibody against COP10, so she could see in which fractions COP10 was enriched. And after that, she collected those fractions and separate them in a native gel. You can see here, so in that native gel, she could still see two different bands that she sliced those bands, elute proteins from there, and then separate them in a denaturing gel, no, by SDS page. So here you have the lower fraction and the upper fraction. And actually the upper fraction here, so the upper band, contained COP10, but also different specific bands here, and that she could actually slice and send for mass spectrometry. And she could identify the components in that complex containing DDB1A and DIT1, also COP10. Actually, DIT1 was kind of truncated, so she could find different bands for DIT1, okay? So what are the disadvantages of this system? So, well, one of them is that it requires a deep knowledge of protein biochemistry. Second one, it's kind of relies a lot in trial and error. So you have to try different purification steps to see whether you indeed get an enrichment of your protein or you lose it. So it's kind of also a bit blind. And also, as you could see, it's prone to degradation because there are many purification steps in which you can lose your proteins or proteins of interest. And also, as I said, so, well, it requires a large amount of material to start off. And indeed, Yuki, purified this protein complex using 1 kilogram of fresh cauliflower. Actually, she was using the outer layers of cauliflower, so you can imagine the smell that was in the lab at that time. So, and actually, it was really painful because to see her so working for many and many days, just slicing the outer layers of those cauliflowers to end up purifying that complex. So that's why other approaches are more favorable or preferable, so to carry out the purification of specific protein complex. And this is what we want when we come to the affinity based technologies to purify protein complexes. So the typical ones are those that are based on a single epitope, no? That it's the ones that you know that are usually used for co-immunoprecipitation assays, no? In which we fuse our protein of interest to a specific epitope, no? HA, FAC, there are many different flavors for those. And also the ones that are, I mean, that we have been working a lot with is those that are based in the fusion of your protein of interest to two different epitopes. So we call it, which allow the tandem affinity purification by two different steps of affinity purification. And in the middle, there is a step of elution by cleavage with a specific protease. No, I will go into details here. So about one step affinity purification. So it's kind of simple. You get your protein extract. Here you have to pay attention to some specific considerations while preparing your buffer. So you want to keep your proteins as much as possible under native conditions. So you need to use buffers, that kind of you use mild conditions for incubation of the proteins with a specific affinity matrix or resin, and that has affinity for that epitope that is fused to your protein. Then you will use different washing conditions to remove all contaminants. And finally, you will use elution. So, and the idea is that upon elution, you will get your protein bound to all those interacting proteins that are in your cell-specific type or tissue. So this system, together with mass spectrometry, can allow identification of proteins that are bound to your specific protein, but also, as I said, it allows co-immunoprecipitation assays in which we can, for example, analyze the strength of a specific interaction or protein-protein interaction. No? Here, for example, this is from one study in which I participated a while ago, carried by Yusuke Saijo. So, so he was trying to characterize the interaction of COP1 with phytochrome A, and well, he was pulling down COP1 using an antibody against COP1. And here you can see the amounts of protein that he was able to immunoprecipitate. And together with COP1, he could detect a partner of COP1, which is SPA1. So he could have a relative idea of the amount of protein that he was co-purifying, so how well the process was going on. And as a readout, he was checking phytochrome A, but actually he was not checking just unmodified phytochrome A, but also phosphorylated phytochrome A. So under different genetic backgrounds, so he could identify which one was the preferred one to interact with COP1. Here you have all the relative quantifications of the COP1 interacting with SPA1, but also with phosphorylated or unphosphorylated or unmodified phytochrome A, depending on whether these other light signaling components were present or not. No? So it means that the co-immunoprecipitation of proteins can be kind of powerful to analyze the kinetics or the dynamics of a protein complex assembly with different isoforms for a specific protein. So then this is the other one that we use in the lab. I mean, in a daily basis, which is the tandem affinity purification of protein complexes, in which this is the original system that was set up in yeast. So this was about 2001, I think, by Gavin and collaborators using yeast. So actually they used this system. So originally in yeast to characterize the interactome of yeast. And this was actually a milestone, no, in the study of proteomics, by setting up this technique that afterwards was transferred to other biological systems. So in my case, I try to apply to the identification of protein complexes in Arabidopsis or other plant systems. And here what you will do is to fuse your protein of interest to different types, to different epitopes. And the most common ones, so well, after first affinity purification step, using one of the epitopes, usually the IgG binding domain. So, and after washing, you can elute your proteins by cleavage with a specific protease for which you have a target site in your epitope. And, And then there is a second affinity step, in this case using calmodulin beads. And you can elute your proteins using EDTA or EGTA, no? Because this is based in this interaction requires cations. So after elution, you can analyze the composition of those protein complexes by mass spectrometry. So different systems were developed a few years ago for tap purification in plants. The one that I developed is around here, which we call the alternative tandem affinity purification system, in which we use two IgG binding domains, and we have not two epitopes, but also we have two different epitopes that allow different alternatives for a second affinity purification step. So 6 histidines, no, to be purified in a nickel column, or 9 repeats of the MIC epitope, no, that could allow identification of your protein by using an anti-MIC antibodies. No? So this was the system that I developed at that time. So in which we would express different fusions, could be N-terminal fusions to the tap epitope or C-terminal fusions to our protein of interest. This was based on the cloning system was based on gateway, so which would facilitate cloning of multiple proteins. So and to carry out interactomics, no, with a large number of proteins. And we decide to use as a system to test this Arabidopsis, no, right away in planta. And the system worked at the time, so this was the proof. So we purify the COP9 signalosome by using this alternative tandem affinity purification system. So here you can see, and upon elution of proteins, so this is a silver stained gel, you can see all these different bands that correspond to all different subunits. So CSN1, CSN4, 5, 6, 8 subunits in the COP9 signalosome, detected also by Western blot. And at that time, we are talking about 2005. So this was still kind of challenging, because to send this protein for mass spectrometry, so you were requested to send something that was visible by Kumasi staining. So we are talking about micrograms of protein. So this was a nightmare to identify proteins using that system from Arabidopsis. So you usually had to use about 50 grams of material to get one of these gels. You can imagine to get it in Kumasi, so maybe 10 times more. So, and also there was an issue, so that we were really worried about that at that time. So whether the fusions were functional. So the epitope was kind of large, and when we fused it to our proteins, so in most cases we could see partial complementation of the mutant phenotype. So this was the functional analysis that we were carrying out, to test whether the resulting fusion protein was able to complement or not a specific mutation. No. Here you can see for high 5 mutant, because we were very interested in photomorphogenesis, so that there was partial complementation of the phenotype. So the high 5 mutant has an elongated hypocotyl compared to the wild type. And here you can see that the complementation was intermediate. I mean, the phenotype of the complementary lines was intermediate. So, and actually it depended on the wavelength. So for example, for under red light conditions or blue light conditions, the complementation was much better than we were using far red light conditions. So this was an issue at that time that we thought that it could limit the use of this system, but indeed, so we decided to go, I mean, to keep going and to use this system to purify the protein complexes and see what we were able to interact, I mean, to identify as interactors of high five. So, and actually afterwards, so all those limitations as I was talking about, so the amount of protein to be used, the limitations based on mass spectrometry of the efficiency in the detection of proteins by mass spectrometry, and these functional issues I was talking about, so actually were somehow overcome by an improved system of Tandem affinity purification developed by the lab of Gert Jager in the VIB in Ghent, in which what they did is to use a different tab epitope, much smaller, still using the IgG binding domain. For the second epitope, they use the streptavidin binding protein, which is kind of smaller. And for the cleavage, they use the 3C protease from rhinovirus, which is active at 4 degrees. And this is pretty nice because it can allow to carry out all purification steps under cold conditions, so which stabilizes the protein complexes and impairs their degradation. So they have generated a collection, a large collection actually of different constructs using what they call this GS rhino tag, no? Based on the IgG and streptavidin binding protein and the rhinovirus 3C protease for fusions in the N-terminus and the C-terminus. This is also a consideration, so that this needs to be taken into account. So when you are working with a specific protein and you don't know, I mean, whether your protein is going to be functional or not, if you produce N or C-terminal fusions, you should check both. So unless you have functional data or evidence showing that the C-terminal or the N-terminal one is the appropriate one, or is the one that's going to be functional. So if not, usually we make fusions to the 2 different versions and produce the 2 of them and see whether the 2 of them are really readily pulling down proteins or whether those potential interactors are meaningful or not. So, and we have found actually that this is important. So in some cases, just by making a fusion to wrong side of the protein, so can impair completely the interaction with other proteins, because maybe it's actually blocking the formation of the right complex, no, of that protein. And also that tagging in the wrong side can block a specific subcellular compartment with a transit peptide, for example, a nuclear localization signal. So that could actually impair the function of the protein and the ability to interact with the right proteins, no, in the plant cell. So this is also based on gateway. And another one of the advantages of this system is that they use cell cultures. And actually this allows purification of protein complexes in a scalable manner. So actually you can scale up these cultures to get grams of material. These are Arabidopsis PSBD cells, actually, that you maintain shaking them in specific temperature conditions, and that we maintain them also under dark ground conditions. And this is because we want to avoid production of Rubisco, which is a, say, large contaminant in the purification of proteins from plants. And here you have two different examples. So this is the healthy one. When we see this culture in a flask, so we say, okay, so this has the green light for purification. And usually we have several replicates for purification. And as Monica will show you later, when she will start showing you how to filter and analyze the material, so she will use two different replicates from a specific bait. And indeed, so this is the process. So just quickly, so just a reminder. So we have a first purification step using IgG beads that allow binding of our protein complex to the IgG binding domain. Then we have some washing and a cleavage by the 3C protease, which is commercially available. And afterwards, second affinity step, using in this case tabidine beads that will bind to our tabidine binding protein in our tab tag. And finally, we can elute. And the elution, we usually do it under denaturing conditions. Actually, we use an SDS page loading buffer. So just boiling and using very strong denaturing conditions to remove all proteins from the beads. And this is what we will send for mass spectrometry. So actually, before that, so those proteins will be, oops, sorry, second. Okay, so sorry for the interruption. And those proteins must be digested. Usually the typical enzyme to do so is trypsin. And there are, there are different equipments to carry out that process in a high throughput manner. Okay, so after actually, okay, sorry again. Once those proteins can be digested in gel, so actually what we will do is to load those proteins in a SDS page gel, and the bands will be sliced and digested in gel with trypsin, and proteins will be eluted from the acrylamide bands and injected upon elution. They can be injected to the mass spectrometer. So this is I will go also quickly through this part, because I mean we are not experts in the mass spec itself, so we are users. So as I said, proteins from the SDS page gel can be sliced, digested in gel, eluted, and what we will get upon mass spectrometry is a fingerprint of that protein based on the spectra of peptides collected, and which we call the peptide mass fingerprinting. And as I said, there is a drawback here, is that some proteins can be missed or lost, particularly those that are low abundant. Okay, and there are many different equipments for mass spectrometry. Usually they are based on 3 different devices. So the ionization device, the mass analyzer, and the detector. So, and we can classify these equipments depending on these 3 devices, which is a MALDI-TOF or what is a QTOF. And about the ionizing, so the first device. So there are usually 2 different types. The most commonly used are these 2 different types. One of these they call, which is called the MALDI, which is matrix assisted laser disruption ionization, and the other one is the Electrospray ionization. Then we have the device that allows separation of proteins depending on the mass to charge ratio, or the mass analysis, so which is depending on the time of flight of those peptides, or those that are based in quadrupole mass analyzers, and also those that are based on ion traps. For example, the Orbitrap, which is actually the so-called the Ferrari of mass spec equipments. And just some considerations about MALDI. MALDI, what you do is to fix your protein sample into specific matrix that will be ionized upon, using a laser, and this will allow emission by heating of those peptides and their vaporization and actually their ionization. And that can be, well, here it says that can be usually, used right away from protein samples from 2D gels. And this would be the picture of what I was explaining. So here what you have is that matrix that contains our protein mixture digested with trypsin. So here we have peptides, specific peptides for those proteins, and upon laser irradiation, where we get this heating and ionization of the peptides that will go through electric field, and those proteins will be separated depending on the time of flight. That time of flight depends on the mass to charge ratio. So actually, those proteins will take some time, to fly from the ionizer to the detector, and we can separate them and analyze it. Then those peptides, depending on their time of flight, we can discriminate them classify them depending on their time of flight. And that time of flight is a specific, let's say, of each specific peptide. So you get this kind of spectra of different peptides that have been separated depending on the M2C ratio, the mass to charge ratio, and depending on these numbers, so we can, this MC ratio, so this is a signature for each specific peptide, and we can identify them and to the protein that they correspond to. Okay, so then we have this other ionizing system, which is the Electrospray, and here, well, what we'll do is to pass our sample through a capillary tip to become an aerosol that can be also ionized using electric field, and you get a cloud of ionized peptides here, better to see in the picture. So here you see this capillary here with our protein mixture in a specific liquid matrix. And what we do is to vaporize it and in small droplets that can be ionized, and well, actually they will go through electric field again. And here what we used to is which is called a quadrupole that will produce a variable electric field that, depending on the physical chemical properties of those specific peptides, will allow separation of them. And then afterwards we have again a detector that will receive those peptides depending on their separation in this quadrupole mass filter. So this is a very simple view of the system. So I think at the user level is enough. And usually these mass spectrometry system are carried out in tandem, and it's called MSMS, and it's basically based on the fractionation of those specific peptides. So which you get after the collection of those peptides is to fragment them one by one and to get another spectra for each peptide depending on their fragments, and that allows better or more reliable identification of each specific peptide. Okay, so once we have those spectra of the collection of peptides obtained from our samples, this is what you get indeed. So this is what the user gets. It's a list of potential proteins that have been searched using different engines. So the typical one is Mascot. It's also sequenced depending on the the company, the seller of the mass spec. So they have different engines, to carry out these searches. So Mascot is I guess the most common one. Maybe someone could kill me for selling this. Well, this is the one that we use in the lab here. These are the different parameters that you get in the list of proteins. So this is not Arabidopsis, as you can see. So this is an example. And here you get, as I said, different parameters. So the database in which you make your query and the number of entries on that database. So the proteins that are represented in that database and the number of residues of amino acids that are in there. Here is the enzyme that we have used, trypsin. This is important consideration because depending on the enzyme, the protease that you use to cleave your samples, you will get a different collection of peptides, and different databases could have been produced using different enzymes. So this is something that you have to take care of. Then also modifications on the peptides during the process, particularly oxidation or methylations, different search parameters so that we can define the mass error in the MS or MSMS spectra. And here you get this list of your proteins ranked depending on the protein score. And for each of those proteins, if you click on them, you get different parameters depending on the mass of the protein, the expected mass. This is in kilodaltons, so Dalton, sorry. A protein score that is assigned by the search engine, depending on an algorithm, and that score will depend on the quality and the quantity of the peptides that have been identified. So the more peptides you identify, the more reliable the identification is. And if those peptides are more specific for that particular protein, so the score is higher. So if that specific peptide is just found in your in this specific protein, so that increases the score, of course. And here you have the number of peptides that have been identified and the total number of unique sequences identified for that specific protein in your spectra. And here, what you have down here, I guess, yeah, it's other proteins that could have the same collection of peptides. So this identification, well, is reliable, but also, is not definitive because other proteins in these organisms is sharing the same number of peptides. Here is the Mascot score for each specific peptide. And actually the score for a protein is derived from the score for each peptide. So it's according to the algorithm used by Mascot. So this is what we will get in our list. And in the training, in the hands-on part, so Monica will show you how to analyze and filter and see which proteins are significant or not, depending to different parameters, so the protein score, the number of peptides identified, and also she will carry the filtering against the background list of proteins, in this case of Arabidopsis, so which we call the crapon. So this is an overview of what we get. So we fuse our protein to a specific tag. Then we express it in cell culture to get a large amount of protein. Actually, I didn't say that, but this Arabidopsis cell culture, particularly the PSBD, is an octaploid. So you get a huge amount of your protein. So the levels of expression are pretty high. Then you get the two affinity purification steps: elution, the mass spectrometry, the identification of peptides, and the identification of the corresponding proteins, a collection of proteins. After the filtering, so what you can get is a number of proteins that interact with your protein of interest. So here, as an example, because it's the protein that I will use for the second part of my talk, is DDA1, for which we could identify all these components in the complex. We could identify DDB1, which is actually the substrate adapter that connects DDA1 to the rest of this E3 ubiquitin ligase. It's a Calin-4 based E3 ubiquitin ligase, but also we could detect COP10, DIT1, and DCAF proteins. So Okay, so a large number of proteins and some additional ones. Okay, so once we have these interactions, so or this potential list of interactors, these are, I mean, in our case, in this case, they are all well-known interactors or well-known members or components of this protein complex. But you can find many other proteins that need to be tested to check whether they are real or not proteins associated to your protein of interest. And for that, again, there are many different techniques. I will just go quickly with a few examples of them, and then I will move to the case of a study, which is actually, for me, the fun part. So, well, as I said, you can confirm these interactions by yeast to hybrid. So, I mean, to check whether there is a direct interaction of your protein with any of the components in that protein complex identified. And this is an example of our lab in which we use different truncated versions for our protein of interest to see which domains interact with those potential interactors. And in that case, this protein is Alix, it's a protein is conserved in many different systems, and even in mammalian systems is also present, and fungi. And this protein is related to the vesicle trafficking, and what we could see is that it was interacting with ABR receptors. And we truncated the protein, different fragments, and we could see that this protein was interacting through different domains with the ABR receptors, which you can see here. So we can analyze or characterize which domains interact with each other. Another approach we use in the lab is in vitro and semi-in vivo pull downs. For the first one, what we do is to express and purify specific protein fusions from bacteria. And once they are purified, we mobilize one of them in a resin, in this case, Alix, to the maltose binding protein, and we incubate them with the different proteins purified from bacteria fused to the his tag. And then after washing and elution, we can see which proteins were present or not, whether there was interaction compared to the tag alone, in this case, MVP. Here you can see some background. This is in vitro. So usually what we want to test is whether the interaction occurs in vivo. So here there is a semi-in vivo in which we have a recombinant protein purified from bacteria again, but here we have incubated with a protein extract from plants. Okay, in this case from Arabidopsis, expressing PIL4 fused to the H8 epitope. And here you can see again that we have interaction. As I said, the best thing is to test it in vivo. And for that, there are different systems that allow interaction in planta. One of them is the split luciferase. They are based more or less in the same systems, in which what you do is to split the reporter protein in two and fuse each half to each of the interesting proteins. Protein 1 and protein 2. One of them will carry the N-terminal part of the luciferase in this case, and the other one the C-terminal part. And what you can do is to express these fusion proteins in a specific cell or plant cell system. It could be Nicotiana benthamiana epidermal cells, onion epidermal cells, or it could be protoplasts. And you can use agroinfiltration, or in other cases you use biolistic to introduce these protein constructs or actually what you transform is the plasmids that will express the proteins, and what you try to see is whether there is reconstitution of the luciferase activity, and upon adding the substrate, in this case luciferin, you will see a signal if there is interaction of the production of luminescence due to the catalysis of luciferin. And this is what you can see here in an example of a paper in which we collaborate actually with HD2C and host 15. So this reminds me the talk of Marta. And here, well, this group, our collaborators, were characterizing the function of a host 15, which is a member of Calin-4B3 ligases, the ones that are of our interest. And they found interaction with this histone deacetylase, HD2C. In planta here, by reconstitution of the luciferase activity. Here in this case is by agroinfiltration, but as I said, other alternatives is like expression in protoplast. And you can actually quantify the strength of that interaction, no? But depending on the luminescence that is produced. Another system, which is related to the other one, is the biomolecular complementation assays, in which the reconstitution is for fluorescent protein, GFP, YFP, or reported fluorescent proteins. No? Here you fuse one of the proteins of interest to the half or a portion of the YFP in this case, and the other one to the other half. And here you can see that upon our infiltration of those constructs into Nicotiana mentamiana leaves, we could see interaction of the proteins in nuclei. Actually, they are staying here with DAPI, and this is our positive control. And well, you can do some measurements of the interaction of this, of the strength of this interaction, depending. I mean, what we did here is to count the number of nuclei in which we could see fluorescence. No, is a bit painful, but can provide you with some sort of measurement of the reliability of that interaction. And actually, we have used this technique to not just check for binary interactions, but also for formation of trimeric complexes. And this is an old studies already in which we were characterizing the interaction of COP1 with other proteins controlling the circadian rhythms in Arabidopsis, including L3 and Gigantea. And we had evidence that COP1 and L3 were interacting here in speckles, in nuclei. So you can see here the reconstitution of the fluorescence by interaction of these two proteins. Also of L3 and Gigantea here. And then we check whether COP1 and Gigantea were themselves interacting. And we couldn't see any signal. But when we are going to infiltrate the three proteins together, so these two fuse to portions of the YFP and the other one that was targeted with HA, if I remember well. So then is when we could see reconstitution of the complex. So L3 was allowing interaction of the three proteins, okay? So of the Gigantea and COP1, and was necessary for that interaction. So, and then finally, so the typical ones is to check by co-immunoprecipitation. So just to introduce some of the lines that we use in the lab, as you will see later. So we have plants that express DDA1, a component of these complexes. And in Arabidopsis, expressing also COP10, another component of the complex, and by pull down, we could see interaction. I mean, by co-immunoprecipitation, we could see interaction of the two proteins in the same complex, even though they are not directly interacting. They are part of the complex. They are at different sides of the protein complex, but they come together. So this is nice to test for interactions that are not direct, to test whether two proteins are in the same complex, even though they are not directly interacting with each other. There is association in the same complex. And here we have for DDA1 and Calin4, that again, there is no physical interaction between DDA1 and Calin4, but they are part of the same complex. So in this way, we can confirm interactions obtained by TAP, that don't rely in binary interactions. And well, so this is the last slide of the first part. I guess I have about 20 minutes more, for the second part. And well, I should go faster because some questions might be.
Yes, there are some questions already. So just if you will go through the conclusion, maybe you could answer the question and then go for the case study part, if it's okay with you.
Okay. Well, so yes, the conclusions is mainly based on the TAP. So, I mean, it's widely used for protein-protein interaction and protein complex composition analysis. Is fast, is simple, and produces is very reliable, and you can get a high yield of your protein complex. And particularly when we use Arabidopsis suspension cultures, is really high throughput. And but it can be used also in other plant systems. So it has been applied also to Nicotiana, Medicago, rice, maize, and we are working now setting up the system in Mercantia. And well, using the system, you can actually purify proteins that stably or transiently associate with your protein complex. So saying that, so actually, well, there could be some issues about the functionality of the proteins when expressing in radiopsis cell suspension cultures, because actually you don't know whether they are fully functional, okay? And also whether it is necessary or not to carry out the 2 purification steps. So actually has been proven is that just one single step can work nicely, because the mass spec techniques have been improved so much that they are able to analyze very complex mixtures of proteins. So you can identify a large number of proteins just by a single step of purification. But again, so you will need a nice filtering about, to remove all the contaminants from those. And that removal of contaminants depends a lot of having a nice collection of contaminants obtained upon purification of many different protein complexes, as you will see later. And okay, so this is the end of the first part. So for questions, how do I do? Should I read them somewhere, or?
Sorry, I was muted. So yes, we have them on chat, but maybe Katerina and Maria could ask them, could just ask the questions by themselves. But in case you will not hear it, they are on chat.
Okay. Thank you for a really nice talk. I am wondering if you blocked during